Good morning, everyone. Welcome to today's presentation. I'm Dr. S.K. Singh, and I'm excited to share with you some insights on moist heat sterilization, specifically steam sterilization. In this session, we'll explore how this method is used to ensure safety and sterility in various settings. We'll cover the principles, procedures, and key considerations for effective sterilization. Let's get started. Let's start by understanding what moist heat sterilization actually is. It's a process that uses saturated steam under pressure to effectively destroy microorganisms, including spores, which are often the most resistant forms. This method is by far the most widely used in the pharmaceutical industry because of its reliability and efficiency. It's particularly preferred when the materials being sterilized are stable under heat and moisture, ensuring they won't be damaged during the process. Now, moving on to the principle behind it. The key idea is that steam transfers heat rapidly and efficiently to the items being sterilized. This heat causes proteins within microorganisms to denature and coagulate, effectively killing them. But for this process to work properly, we need several critical factors in place. These include the right temperature, sufficient exposure time, adequate pressure, and high-quality steam. One of the most important concepts to remember is the idea of the cold spot. That's the part of the load that's the hardest to sterilize because it's the coolest point, often in the most insulated or deepest area. Ensuring that the cold spot reaches the necessary sterilization conditions is what ultimately determines whether the sterilization process is successful. So, in essence, understanding and controlling these elements is vital for achieving reliable sterilization in pharmaceutical settings. Let's take a closer look at a typical sterilization cycle. Usually, you'll see it either at 121 degrees C for about 15 minutes or at 134 degrees C for a shorter period, around 3 to 5 minutes. Now, it's important to remember that these are just standard examples. The actual cycle you use isn't universal. It really depends on several factors. For instance, the type of load you're sterilizing, the packaging, and the volume all influence the cycle parameters. Plus, the specific equipment you have also plays a significant role. Speaking of equipment, let's talk about autoclaves or steam sterilizers. There are different types to consider. You might be using a gravity displacement autoclave, which is quite common, or a pre-vacuum type that offers faster and more efficient sterilization. There's also the liquid cycle autoclave, which is tailored for sterilizing liquids. Now, regardless of the type, autoclaves have some key components. The main chamber is where the sterilization happens. Then there's the steam generator, which produces the sterilizing steam. And finally, sensors. These are critical. They monitor temperature and pressure throughout the cycle to ensure everything is proceeding correctly and safely. All these elements work together to achieve effective sterilization, which is essential for maintaining safety and compliance in our practices. All right, let's walk through the step-by-step method for sterilization. First, loading is crucial. You want to ensure items are placed properly so steam can penetrate effectively. Proper arrangement means no overlapping or blocking of airflow, which helps achieve uniform sterilization. Next, we move to air removal. It's essential to get rid of air pockets, either through vacuum or displacement, to allow the steam to contact all surfaces thoroughly. Once the air is out, we proceed to heating. The goal here is to reach the required temperature throughout the load. It's important to remember that the entire load must reach this temperature before the sterilization timer starts. Only then can we confidently move into the holding or sterilization phase, where maintaining that temperature for the right amount of time is key. Finally, after sterilization, the cycle concludes with cooling and drying. This helps prevent contamination and ensures items are safe and ready for use. Following these steps carefully ensures the entire load is properly sterilized, which ties back to our overall theme of maintaining the highest standards of cleanliness and safety. Let's take a closer look at the validation requirements. Validation is all about ensuring process reliability, so we need to systematically confirm that our procedures consistently meet the desired standards. This involves several key steps, starting with IQ, which stands for Installation Qualification. This verifies that all equipment and systems are installed correctly and according to specifications. Then there's OQ, or operational qualification, which tests that the equipment functions properly within its intended operational ranges. Finally, PQ, or performance qualification, confirms that the process consistently produces the desired results in real-world conditions. Now, in addition to these qualification steps, there are specific studies we need to conduct. These include heat distribution studies to ensure uniformity, heat penetration studies to verify that heat reaches all parts effectively, and biological indicator studies to confirm sterilization efficacy. We also have to adhere to strict regulatory requirements, like GMP and WHO standards. It's essential to follow guidelines such as WHO GMP, EU GMP Annex 1, and the FDA or EMA guidelines to stay compliant and ensure safety. Moving on to the key expectations, validation is mandatory for these processes. For example, the sterilization process must achieve a sterility assurance level, or SAL, of 10 to the -6, meaning there's a very high assurance that the product is sterile. We also need to monitor temperature and pressure throughout the process continuously. To do this accurately, independent probes are required. These help ensure the measurements are unbiased and reliable. All these elements come together to build a robust, compliant, and reliable sterilization process that supports the overall quality and safety of our products. Let's talk about biological indicators, which are a crucial part of ensuring sterilization effectiveness. The organism we typically use is Geobacillus sterithermophilus. This particular spore former is known for its high resistance, meaning it's tough to kill, so if it's eliminated, we can be confident that the sterilization process is effective against most microorganisms. We use these biological indicators to confirm lethality, essentially, to verify that our sterilization process has successfully destroyed all living organisms. The key point here is acceptance criteria. After the sterilization cycle, if there's no growth of these spores after incubation, we consider the process successful. Now, to reliably achieve this, we monitor several critical process parameters, or CPPs. These include temperature, because it needs to be high enough to kill spores; time, ensuring the process lasts long enough; pressure, which supports the sterilization environment; and steam quality, since the purity and moisture content of the steam directly impact its effectiveness. But it's not just about the sterilizer settings. Load configuration plays a role too. How the items are arranged can affect sterilant penetration. And air removal efficiency is essential because trapped air can prevent steam from reaching all surfaces, reducing sterilization efficacy. So, in summary, maintaining and verifying all these factors ensures the sterilization process consistently achieves the desired level of microbial kill, safeguarding product safety and quality. Now, let's take a look at some of the disadvantages associated with this sterilization method. First, it's not suitable for heat-sensitive materials. So, if your items can't withstand high temperatures, this might not be the best choice. Similarly, products that are moisture-sensitive could also be at risk, as excess moisture can cause damage or compromise their integrity. We also need to be aware of certain risk factors that can affect the effectiveness of the process. Improper air removal can leave behind pockets of air, which prevent the sterilant from reaching all surfaces. Load configuration issues are another concern. If items are packed incorrectly, it can hinder penetration. Wet loads are also problematic because moisture can interfere with the sterilization process and potentially damage the products. Now, despite these disadvantages, it's important to remember the advantages that make this method so popular. It's highly effective, really excellent at killing spores and ensuring sterilization. Plus, it's non-toxic and eco-friendly, which is a big plus for sustainability and safety. It's also fast and economical, making it efficient for many settings. The method allows good penetration of steam, ensuring thorough sterilization even in complex loads. And importantly, it's the regulatory preferred method in many industries, which underscores its reliability and acceptance in professional standards. Now, let's take a look at some of the key applications of this material. It's quite versatile and plays a crucial role in various fields. For example, It's used in the preparation of culture media, which is essential for growing and studying microorganisms in laboratories. This highlights its importance in microbiology and medical research. Additionally, it's utilized in the manufacturing of surgical instruments, where its durability and stability are vital for patient safety. You'll also find it in aqueous injections, serving as a reliable component in pharmaceutical formulations. Furthermore, it's commonly used for glassware, providing strength and chemical resistance, which is critical in laboratories. Lastly, it's employed in rubber closures, ensuring secure sealing for vials and bottles, which helps maintain the sterility and integrity of medicinal products. Overall, these applications emphasize how essential this material is across healthcare, research, and manufacturing sectors, supporting safety, effectiveness, and scientific advancement. All right, let's walk through this example calculation. We start with a temperature of 121.7 degrees Celsius and a process time of 15 minutes. The key here is to determine the lethality factor, which tells us how effective our process is at inactivating microorganisms. So we calculate the lethality factor using this formula: 10 raised to the power of 121.7 minus 121 divided by 10. When we do that math, it comes out to approximately 1.1487. This number essentially adjusts the process time based on the temperature. Next, we multiply this lethality factor by the process time, 15 minutes, to get the equivalent F0 value. That results in about 17.2 minutes. And here's the important part. Since our F0 value is 17.2 minutes, it clearly exceeds the required minimum of 12 minutes. This means our process meets the necessary sterilization standards. This example highlights how we can quantify and verify the effectiveness of our thermal process, ensuring safety and compliance. Let's conclude by emphasizing that moist heat sterilization, or steam sterilization, stands out as the most reliable and widely accepted method in pharmaceutical manufacturing. It's highly effective because it can achieve a very high level of sterility assurance, specifically a SAL of 10 to the minus 6, primarily through the process of protein denaturation. This method depends on saturated steam, which provides the heat, and on proper removal of air to ensure uniform heat distribution. It also requires validated control over temperature, pressure, and time to guarantee consistent results. Some key advantages are its high efficiency, non-toxicity, cost-effectiveness, and strong regulatory acceptance. These factors make it the preferred choice in many settings. However, it does have limitations. It's not suitable for materials that are sensitive to heat or moisture, and maintaining strict process controls is vital to prevent issues like wet loads or incomplete sterilization. Overall, when validated and monitored correctly, according to GMP and WHO guidelines, moist heat sterilization remains the gold standard for sterilizing heat-stable pharmaceutical products and equipment. It's a cornerstone of our sterilization practices because of its proven reliability and effectiveness. For further support, please feel free to contact us. We're here to help you with any questions or additional information you might need. Thank you so much for your time and attention today. We truly appreciate your interest and engagement. If you require any assistance moving forward, don't hesitate to reach out. Again, thank you and have a great day.